Welcome everyone. Here we'll discuss the C4 pathway. In the previous class we have discussed about the C3 pathway. Now in today's class we will discuss about the C4 pathway. Okay, and here C4 pathway, it takes place in C4 plants, that is the plants where the first stable compound which is formed when carbon dioxide is fixed is a 4 carbon compound, and that 4 carbon compound is that is oxaloacetic acid. Okay, and we have also seen that there are special arrangements which are there in the anatomy of the leaves of C4 plants. They show Cran's anatomy, that is wreath like arrangement of bundle sheath cells around this vascular bundle. And apart from that, they tolerate very high temperatures, they show that is response to very high light intensities, and they lack photorespiration, and because of that the production of molecules, that is biomass, is very high. Now let us see how the C4 cycle takes place. How the C4 cycle takes place. Here the C4 cycle is also called as, or the C4 pathway is also called as Hatch-Slack pathway. It is also called as Hatch-Slack pathway. C4 pathway is also called as Hatch-Slack pathway. Okay, in this let us see what happens. Here now the cycle takes place in two different cells. Here in this let us see this circular, that is the box which I have drawn. So this is the mesophyll cell. This is the mesophyll cell, and this is the bundle sheath cell. This is the bundle sheath cell. Okay, and here in plants we have, this is the inner cell, that is plasma membrane, and this is the cell wall. Here also this is the cell wall and this is the plasma membrane, because in plants the plant cell is covered by two, that is there is an extra addition of membrane, that is the cell wall, which will be present in the plants. So in animal cells there is only plasma membrane. So but as of now here this is a cell, this is one cell. Which cell it is? Mesophyll cell, and this is a bundle sheath cell. This is a bundle sheath cell, and here there is channel is there. This channel is called as plasmodesmata. The channel is called as plasmodesmata. So one cell is connected with the another cell with the plasmodesmata. So what happens here in whichever the mesophyll cells? This is a mesophyll cell and this is a bundle sheath cell. So we are talking about C4 pathway. In C4 pathway, that is C4 pathway or the Hatch-Slack pathway, it takes place in C4 plants, and in C4 plants we find this bundle sheath cells. These are in present, that is they are present in the surroundings. For example, if this is the leaf, this is the palisade parenchyma. Okay, here is the vascular bundle and here will be your bundle sheath cells. Here will be your bundle sheath cells. Let us examine this. This cell will take and this cell will take. Okay, this mesophyll cell and this bundle sheath cell. So this is this mesophyll cell and this is this bundle sheath cell. Okay, mesophyll cell and bundle sheath cell. So they are connected. Now let us see how the C4 cycle takes place. Okay, in the C4 cycle what happens is the atmospheric carbon dioxide enters into the cell. Okay, the carbon enters into the cell which is taken by this HCO3 in the form of HCO3. The carbon dioxide is taken inside the cell in the form of this HCO3. Here what happens is a 3 carbon compound, that is phosphoenol pyruvate. Phosphoenol pyruvate, it is a 3 carbon compound. It is a 3 carbon compound. This phosphoenol pyruvate, which is a 3 carbon compound, which is already present in the cell. In this mesophyll cell, phosphoenol pyruvate, which is a 3-carbon compound. It will be already present inside the cell. So what happens? This atmospheric carbon dioxide, when it comes, it is taken by this HCO3 in the form of HCO3, atmospheric carbon dioxide. So here carbon dioxide, atmospheric CO2, that is carbon dioxide will be from the atmosphere will enter into this bundle, that is mesophyll cells, okay, in the form of HCO3. Now already a 3-carbon compound will be present. What is this 3-carbon compound? Here that is phosphoenol pyruvate. This 3-carbon compound is called as phosphoenol pyruvate. It will be already present in the cell. So this 3-carbon compound, that is phosphoenol pyruvate or PEP, it is also called as PEP, phosphoenol pyruvate, okay, mixes with this carbon dioxide, combines, not mixes, this phosphoenol pyruvate combines with this carbon dioxide which is present in HCO3. Okay, now here carbon dioxide is fixed. The carbon dioxide from this HCO3 is fixed with this phosphoenol pyruvate. It is fixed with phosphoenol pyruvate, which is called as fixation, carbon fixation. Here this carbon from the atmospheric oxygen is fixed, is joined or combined with a 3-carbon compound, that is phosphoenol pyruvate. So when both of them are fixed, when this carbon dioxide is fixed with this phosphoenol pyruvate, so what is formed is a C4 compound. C4 compound. So what we said, in the plants where C4 cycle take place, or in C4 plants, the first stable compound which is formed after carbon dioxide is fixed is a 4-carbon compound. It is a 4-carbon compound. So here when this carbon dioxide is fixed with this phosphoenol pyruvate, it forms a, that is, this process is called as fixation, and when it fixes, when this carbon dioxide is fixed with this phosphoenol pyruvate, what we get is a C4 compound, that is C4 acid, that is nothing but oxaloacetic acid. C4 acid, which is nothing but oxaloacetic acid, and this fixation of this phosphoenol pyruvate with this carbon dioxide is with the help of the enzyme Pep Carboxylase. It is with the help of the enzyme Pep Carboxylase. Here in mesophyll cells they lack rubisco. In C4 plants, in the mesophyll cells, rubisco enzyme is not present. Instead of rubisco enzyme, the enzyme which is found in mesophyll cell is Pep Carboxylase. The enzyme which is found in mesophyll cells of C4 plant is Pep Carboxylase. They lack rubisco. Mesophyll cells lack rubisco. There is no rubisco in mesophyll cells. But in C3 plants, in mesophyll cells, rubisco will be present, which will be combining the carbon dioxide with the RUBP. But here rubisco is not present in C4 cells, mesophyll cells of C4 plants. Instead of rubisco, what is present is the Pep Carboxylase. Pep Carboxylase helps in this fixing of carbon dioxide with this phosphoenol pyruvate, thereby forming a acid which is a 4-carbon compound. It is a 4-carbon compound, and that acid is called as oxaloacetic acid. It is called as oxaloacetic acid. Now, till now this process has taken place in the mesophyll cells. Okay. Now what happens, this oxaloacetic acid will change into another forms, that is aspartic acid, malic acid and all. Then it will be transported. This that is the acid, C4 acid, which is formed in mesophyll cells, will then be transported to bundle sheath cell. So here C4 acid is formed in this mesophyll cell. Now it will be transported. Okay, it will be transported to bundle sheath cell. So the C4 acid oxaloacetic acid which is changed into malic acid, aspartic acid. So that acid C4, all of them are 4 carbon compound only. So that 4 carbon acid, so the 4 carbon compound which is nothing but an acid, it is transported from mesophyll cells to the bundle sheath cell. So here what is the process? This is transported. What is transported? The C4 acid is transported to where? To the bundle sheath cells. Okay. Now here in this bundle sheath cells, what happens is decarboxylation will take place. In bundle sheath cells, decarboxylation will take place. Decarboxylation is nothing but removal of carbon dioxide. Okay, removal of a carbon dioxide. So here how many carbons was there? C4 acid, na? 4 carbon was there, C4 acid. Now here with this C4 acid, from the C4 acid a carbon dioxide molecule is released. Decarboxylation of the C4 acid take place. That is, from the C4 acid a carbon dioxide is removed. From the C4 acid a carbon dioxide will be removed. So from the C4 if we remove one carbon dioxide, so here decarboxylation take place. Here from the C4 acid a carbon dioxide will be removed. A carbon dioxide will be removed. Okay. Now how many carbon compound we have? It is a 3 carbon compound. We have, that is, we have a 3 carbon compound. It was a 4 carbon acid. Now here we have a, that is, acid with 3 carbons. How? Because a carbon dioxide is lost from this C4 acid in the process of decarboxylation. Okay. Now we have a C3 acid. Now we have C3 acid plus along with the C3 acid we have carbon dioxide also. Okay. Now here one carbon dioxide has been taken out. So now we have a C3 acid and we have a carbon dioxide molecule. A C3 acid molecule, a carbon dioxide molecule. Okay. Now the C3 acid will be again transported to mesophyll cells. The C3 acid will be transported to mesophyll cells. First, a 3 carbon compound phosphoenol pyruvate combined with this carbon dioxide and it formed, that is, fixation of this carbon dioxide has taken place with the formation of a C4 acid. The C4 acid has been transported to bundle sheath cells. In this bundle sheath cell, what happened is from the C4 acid a carbon dioxide is released. So now here in this bundle sheath cell we have 2 things, a carbon dioxide, and because from when C4 a carbon dioxide is removed, now it will be a 3 carbon compound. So in bundle sheath cell we have 2 things, one is a C3 acid and another is, if I write, 2 things we have. First is C3 acid and the second is carbon dioxide, and the second is carbon dioxide. So now let us see this first about the C3 acid. The C3 acid, now it will move to this mesophyll cell because it has to be regenerated. This phosphoenol pyruvate has to be regenerated. It is the same. It is acting as the host. It will receive carbon dioxide, make the acid, transport it to this bundle sheath cell, then carbon dioxide will be released, then again the C3 acid will be separated from the carbon dioxide and it will enter into this mesophyll cell so that it can receive another carbon dioxide. So here the C3 acid helps in the regeneration of phosphoenol pyruvate. So what happens? The C3 acid, which is formed by the decarboxylation process, enters into the mesophyll cells. Okay. Now here in the mesophyll cell, the C3 acid is again changed into phosphoenol pyruvate. So here that is the regeneration process. From the C3 acid, phosphoenol pyruvate will be regenerated. So this was about the C3 acid. So this C3 acid has been regenerated into phosphoenol pyruvate. Now the leftover is this carbon dioxide. Leftover is the carbon dioxide. Now this carbon dioxide combines with rubisco. This carbon dioxide combines with rubisco. This carbon dioxide combines with RuBP. The carbon dioxide combines with RuBP, and this combination is because of the enzyme rubisco. It is because of the enzyme rubisco. So rubisco is ribulose biphosphate carboxylase oxygenase. The name of this enzyme, this rubisco. This rubisco joins this carbon dioxide with RuBP. Inside the cell, RuBP will be present in bundle sheet cells, and here pyruvate was there. So now this carbon dioxide combines with RuBP, thereby carbon fixation will take place, and this Calvin cycle will take place, C3 cycle will take place. So that is how even in C4 plants C3 cycle take place. In C3 plants also C3 cycle will, Calvin cycle will take place, and in C4 plants also Calvin cycle will take place. So how this carbon dioxide then combines with a 5 carbon compound, that is RuBP, with the help of the enzyme rubisco, okay, and then from this rubisco, when this it combines with rubisco, triose phosphate will be formed, and from triose phosphate will go reduction, ATP is added and NADPH2 is reduced and all. Then what we get is RuBP will be regenerated and glucose will be formed. So short here glucose will be formed. So the C3 pathway it will enter, which we have already seen the C3 pathway. Okay, C3 cycle or the Calvin cycle we have already seen. Now there are some other plants. This is about C4 plants. There are certain other plants which lives in very hot conditions. Okay, so they undergo a different kind of metabolism, that is called as crassulaceous acid metabolism, that is CAM. We call it as CAM. The CAM pathway. CAM pathway, it takes place on the plants where they live in very arid places, very hot temperatures. Those plants which live in very hot temperatures, apart from the C3, instead of this C3 and C4 cycle, they undergo another pathway, that is CAM pathway. CAM pathway, the full form is crassulaceous acid metabolism. So some plants, instead of undergoing C3 and C4 pathway, some plants in the sense the plants which lives in very high temperatures, so instead of performing the C3 and C4 pathway, they'll perform another pathway, that is CAM pathway. So in this what happens is, it is this CAM pathway is similar to that of C4 pathway. So what happens in in C4 now, here the thing is separated in space in C4 pathway. In C4 pathway, this C4 acid is formed in mesophyll cell. Then it is transported to bundle sheet cell. Inside this bundle sheet cell, the C3 cycle is taking place. So it is separated by space. So it is space in the sense different cells, one cell in another cell. Okay, so one cell is, fixation is in one cell, and then C3 cycle is in another cell. Fixation is in the mesophyll cell and C3 cycle is in the bundle sheet cell. But here in this CAM pathway, what happens is, it is separated in time. During night time, it is separated in time. During night time, carbon dioxide is fixed. During night time, carbon dioxide is fixed, and during daytime they'll make glucose. Here they have another adaptation, that is 2 cells they are using. In first cell acid is formed, and in the second cell it is that acid is decarboxylized and a C3 pathway is taking place. That is space difference, but here time difference. In the night, carbon dioxide is fixed. Okay, in the night carbon dioxide is fixed, and during daytime they produce glucose. So this is about this CAM pathway. Okay, so with this we have finished the C4 pathway as well as C3 pathway. In the next video we will discuss about this photorespiration, which I have already discussed a little, but we will discuss this photorespiration in detail in the next video.